All right, here we go. I'm outside of my usual realm of comfort, just talking about marine mammals in and of themselves. I'm realised over the years I've become very at home just talking about those big old lard balls. But today, Tim gave me a new challenge, which is a very good one. And Tim also gave me the title to my talk, which I quite liked. He said, "What are you going to say?" I'm like, "Oh, give me a title." And so this is what it is, Finding What's Where. And I do think this actually sums up what we've been doing recently quite well. Before I start, I'd like to give my thanks to the people of Ngāti Whātua Ōrākei for having us on their beautiful, beautiful land that we all live in here in Auckland, and I think also the place that is very, very important to so many of so many different kinds of animals that live here in the Gulf. So my talk today is not me. It's a large number of students, mainly here you can see. So we've got Lily Kojman-Ledwood, who's in the crowd. She finished her Masters in 2015. Below her is Ros Putland, who just handed her PhD in, working on acoustics in the Gulf, the Soundscape. In the middle is Craig Radford, who has no photos of him in the field, so this is him. He's probably one of New Zealand's best marine acousticians, and he's based at the Lee Lab. He's a Rutherford Fellow. Olivia Hamilton, who's smiling at the top because she's collecting data. She's on the death throes of writing a PhD and does not look that smiley at the moment. She's currently running distance models and distribution models, so she said she had to dip out today. And then Emma Carroll in the bottom, who was a PhD student with us working on southern right whales and who's now at the University of St Andrews in Scotland on a prestigious Newton Fellowship and a Marie Curie Fellowship. And so this is their work that I present. So when I think of the Hauraki Gulf, I think of the word expansive. It's massive. It's a massive body of water. It's huge. And most of my early work in New Zealand was in the Bay of Islands, which, you know, is much smaller and more cosy, and you're never far from land, but you are often feel very far from land. And as you're moving around this place, you wonder how it is that life actually makes it there. And so when you look at Te Kapuana, Te Moana Nui Atoi, or the Hauraki Gulf, it's a big place, but there's a lot in there. Not only is there a lot of ocean, but there are 50 islands. The marine park itself is around 12,000 square kilometres. It's a lot of ocean, it's a lot of life, and it's a lot of potential. And we've always known that because people have been here for many, many years. The first people came here, and some of them left, and some came back, and some others came. But this has always been a very important place to us as people. But more importantly, it's a very important place to most other life. And thank you, Anne, for saying that about, you know, humans are just one of the many animals, something my daughter is very aware of and has trouble explaining to her friends at school. We're just another animal. So it's a challenge, poor kid, child of scientists. They're doomed. But the other thing that strikes me about the Gulf as well is, even though it is really large, it's ephemeral. You can have these moments in time where you just, you know, will be on your boat or sailing or swimming or flying or whatever it might be, and there's just one little bird. Just one little bird. Like, how can I have been on the water all day and just seeing one little bird hanging around? It's like, dude, where are your mates? I mean, don't know what you're doing. And there's nothing. And then other days, you see this. You know, this magnificent sight, which is a workup. We used to see them. They used to be a lot bigger. Even in my short time working here in the Hauraki Gulf, I've noticed that these workups are smaller and they're shorter. And this is some fantastic footage from the people on Dolphin Explorer, the whale watch boat, who are incredibly generous with our research and researchers, and a lot of what we do is because of them. And if you just wait a minute, all the birds are going because there's the whale. So as a Bruder's whale comes through the middle. And so there were common dolphins there were gannets there, you know, there were gulls there, and there was a very large Bruder's whale coming right through the middle and stealing all the food in one single gulp. They really are a rain on the parade of everything else. But that sort of these moments where there's just so much life in one place, it's almost overwhelming at times. So, you know, we can go from this moving feast, it is literally a moving feast in the Gulf, and from one day to the next, it is a very different place. And I think it's summed up by so much life, because there is, and it starts with this green water. Now, if this was a river, we'd be having a different conversation, but this green is phytoplankton. This is, out in the Gulf. It's way out. It's green, it's rich with life. You take a jar of water out of there and you look at it, and there's so much stuff swimming around in there, it's crazy. There's so much zooplankton. It's an amazing place. And this is where it all starts. This is a highly productive place, a highly productive environment. We know what we cherish about the Gulf. We know what we get out of it and what we think is important. But the thing that struck me when I was sort of, as the Brooders Whale ship strike issue was sort of slowly dying down, I'll talk more about that later, but was that, it's not just about whales, it's about all these other things. And what's important to all of our megafauna, all of those big things, the sharks, the tunas, the whales, the dolphins, and of course the seabirds. What's important to our megafauna? So we decided to survey, sort of broadly speaking, the inner Gulf, and that's about 4,500 square kilometres. So we couldn't swim it, couldn't boat it. We decided to fly it. And the reason why we wanted to get around quickly was to try and survey that whole space in one day, because in 22 hours of boating, that's moving along at about 10 knots or so, survey speed. In 22 hours, it's several days actually on the water, and everything may be different the next day. And what you saw the first half of the survey is different to the second half. And yeah, as Steph said, statistics, big yawn, but man, trust me, when you want to publish your work or get your thesis passed, statistics are very important. So we decided we'd fly it. And this is the transect that we flew. So Lily started this work, her master's thesis, which is an amazing piece of work, formed the pilot for this study. And this is the track that we flew. It takes about 5.5 hours to fly. There are a few random little loopy-loop bits. Those are statistics. And there's a sort of I don't know what that random loopy-loop bit is out off the bottom end there of Aotea, but I think someone wasn't feeling well or something that day. But we got back online and off we went again. So these lines are about 10 kilometres apart, and we flew them over 12 months. We did 22 surveys. We tried to do 2 a month. Weather got in the way for a few of them. We flew at 150 metres above the water, and we flew at 100 knots or 185 kilometres per hour. Now, at that height and that speed, you can actually see a little storm petrel pointing along on the surface of the water. We could see most everything as long as it was near the surface. Of course, some of the water was green, and then it would go out to blue as we further out. But it was, generally the surface life. We saw over 9,000 seabirds from 16 different species. We saw 256 sharks from 5 different species, over 5,000 cetaceans from 5 species, and 5 mobulid rays, the big manta rays, those species. We saw a lot in a little snapshot in time in the Hauraki Gulf. And so what we wanted to then do is take that information and then understand who was where and why. And so we wanted to know things, environmental parameters like distance to shore, depth, substrate. But we also wanted to know some of the biological parameters, such as the distance from fish prey balls and the distance from plankton. So those bottom ones, the red is where aggregations of prey fish were, and then distance from plankton, the brighter blue, is where the plankton were. So we take all of these abiotic layers, the biotic layers, and then we put the species on top. So you can see the top left there, we've got the zooplankton in black, and the fish, the small schooling fishes, the pilchards, saury, jack mackerel, those critical mid-water fishes, and the dots where we cited those. And then the middle figure, we've got bronze whalers in yellow, we've got juvenile hammerheads in the green, and then oceanic sharks in the blue. And those oceanic sharks, we saw a 5-metre dusky shark. They're big sharks out there. Most of them in summer, and most of them near a beach near you. Oh yeah. We saw schools of 30 to 50 hammerhead sharks regularly in the summer months around near Waiheke Island in particular. And they're about 1 to 1.5 metre long schools of these sharks. And we saw she would have been not quite 4-metre hammerhead shark, big female. At the on the right, you've got the Brooders whales in red and the bottlenose dolphins in purple, I think, and then blue are the common dolphins. So you can see the bottlenose dolphins are mostly near the coast, and the Bryde's whales and the common dolphins actually are niche separated. They feed in different parts of the Gulf. So all the dolphins, these big consumers, topwater consumers, have found their place in the Gulf so they can coexist year-round. And on the bottom left, we've got the common diving petrel, and on the right, the gannet. So these are just two examples of the birds. Olivia's going through, she's got 12 species, so we saw 12 species, enough to be able to do this work with. And you can see they too are divided and find their place in the Gulf. And so we were able, for that year of surveys, to take that information about each species. So here is fluttering shearwaters. And we're able to look at the Gulf that has two seasons, effectively. It has the warm season, which is summer and autumn, and it has the cold season, which is winter and spring. And then the darker blue shows those hot spots, those places that are more important at that time for this species. So you can see in February, in the summer, warmer months, the fluttering shearwaters are further out to sea, further sort of out within the Gulf. But come winter, they're further in, they're closer in. And this here for the gannets, a slightly similar picture, but gannets are never too far from their home colony. They don't go too far. They like being in that place. So we can then take the seasonal information, the environmental information, the biological information, what we saw, and then we can put this into predictive models. And so this is one for gannets. So we can actually pull together from all of our data, who do we think is where and why and how might they be there, and what's driving that? And so you can see along the bottom there's a whole bunch of environmental things: chlorophyll A, which is the phytoplankton, depth, distance from 40 metres, distance to the colony, distance to fish balls, distance to plankton, shore, slope, and the sea surface temperature. And these have different effects on the abundance of these birds and the presence or absence of them. And these models are very, very powerful for us as scientists to try and understand how these birds are distributed through time and space and how that might change in the future. And I think this is really important because one of the things that we know in the Gulf is that, food is the thing. That's the thing that links all of these organisms. This is not a reductionist story about seabirds or whales or dolphins. This is about our megafauna who all need the same thing. And so came up with the catchy title of What You Eatin'? And so basically that is my question regularly to these things. What are you eating? Because when you see them, they're doing this. Sure, sometimes you actually see them with a fish in their mouth, or you can see, you're over the top of it, you can see. But oftentimes you'll see kahawai and teraki and things like that, and you know that petrel is not eating a teraki. That just wouldn't work out that well, But, so you see this, but we still often don't know who's eating what. And certainly for those animals, underwater, who spend most of their life feeding underwater, which is certainly the case for the marine mammals, but also for many of the seabirds that dive down and swim under the water, because many of them do, is, what are they eating under there? So I came up with the snappy title of Plankton and Poo. This actually was the title of the grant. It started off as a joke that I could get funding for zooplankton research by filtering it through a whale, which I did. So I got money for whale research. What are the whales eating? But actually, and that's whale poo in the net. It's delicate little stuff. Been eating mysid shrimp, pink poo, mysid shrimps. But then we took plankton samples. And why we did this was because the sort of traditional way of sorting through plankton samples, you do get taxa, but you may know exactly what genus and species, but you don't get many of them. And what we used is high-throughput gene sequencing, new technologies, and we came up with around 2,000 different taxa of zooplankton in the Hauraki Gulf. Now you'll notice here that we've got cool on the left and warm on the right, so there's two seasons, and you'll notice that I've got a big long list of phyla. Why is it phyla? Because our genetic knowledge of the zooplankton taxa throughout the world is really poorly represented in databases, and we need to fix that. But what we know is that there are definite, strong seasonal differences in the zooplankton we see in the Gulf, and there are regional differences. The sort of Firth of Thames area have a very different cohort of zooplankton to the kind of middle Gulf, to the outer Gulf out by Hauturu and out those waters. So we see seasonal and regional differences. And these are really important things to understand, and we need to keep monitoring this because it could detect prey shifts and stability for the megafauna. They are the canaries in the coal mine. If there's not enough food, they won't be there. And this is a long game. And this is where, with modern techniques, that these historical archives and records are valuable for understanding. We can get genetics out of historical specimens and isotopes, things like that. We also wanted to know the Gulf in a different way. What does it sound like? And this is the field of soundscape ecology, and this is Roz's PhD. So on the left side is low frequency, right side's high frequency. So you can see we've got brooders whales, in the middle is fishes, you've got the noisy urchins there in purple in the middle, the snapping shrimp, can't get past the snapping shrimp, and then the dolphins on the far end make high frequency noises. And so we have this rich soundscape in the Hauraki Gulf, and we have a marine dawn chorus. We have a salty dawn chorus. And you can see here at our rocky sites where the hydrophones were set up, that those at that dawn and dusk period, you can see that the little red bars are a little higher up and fuzzy just around the bar, around that dawn dusk. And that is mostly kinna. They're rattling their spines, they're crunching away, but there's a lot of other life. And there is a dawn chorus that we never hear because we're people and we live on top and we bob around in our boats. But you put your head under the water in those places, there's a lot of life. And this makes an acoustic footprint in the Gulf. And we are part of that footprint with shipping, boating noise and like. And that directly impacts on the noises of lower frequency animals like many of the fishes and of course the whales. And you know, one of the interesting side things that's come out of the ship slowing down is that actually their acoustic footprint has been dramatically reduced from about 8 kilometres, I think, down to about 2, which is we didn't realise. But what about the birds? What's their acoustic input to the Gulf? Because you've heard enough of them on land. They're really noisy. So can you turn this up? Yeah. That's the noise of gannets diving. And we expected that. We wanted to know what the sound of a workup was. We wanted to know where the whales and dolphins and other animals using that sound to localise potential workups. And this is the noise of the gannets. But what we didn't anticipate was a completely different noise that is actually a greater contributor. So if you can play. Hear the so there's a dolphin. So that is gannets, all that noisy gannets sitting on the water going to each other as they do, makes a really big noise and it travels a huge distance and the whistles of the dolphins actually attenuate really quickly and we think maybe gannets being noisy are one of the things that is driving the sound of the Gulf. So the Gulf is a place that has changed quite a lot over time since we've been here. We've exploited it heavily since people began. It's an incredible source of food. We are making amends, I think, in lots of ways for this. We're now doing things and thinking about our place in the Gulf and the stories of the animals that live in the Gulf and their history in the future. We've got active management of predators for seabirds, that's critical. We're dealing with a lot of the fisheries bycatch issues, which is hats off entirely to the fishing industry for taking that on. We've got the shipping industry who slowed down. They've done it. I don't need to talk about ship strike anymore because it's done, it's solved, which I think is an incredible success. In 2.5 years, we fixed it. We're now interested in things like nutrient flows and ecosystem flux. We need to be mindful of ongoing bycatch issues because it's not just the fishing industry. And we need to think about things like fisheries for small schooling fishes, those pilchard, saury, etc. They are critical. They are not bait fish. They are not bait fish. They are critical for the big fishes, for the dolphins, for the whales, for the seabirds, for so much life. And it breaks my heart when I see them in a bag of bait. It just breaks my heart. But ultimately, the unicellular thread that binds all of the organisms in the Gulf together are the plankton. 2015-16 saw a massive El Nino event. The whales moved out of the Gulf because the plankton moved out of the Gulf because it was too hot. It was 2 degrees warmer than normal. It was too hot. We will see more of this. And we need to prepare ourselves for this. Everything is going to be affected by climate change in different ways, we don't know. But the resilience of the populations will be challenged and of individuals. Individual animals will be challenged. And we're going to hear from Matt and Brendan in a moment about those lives of individual birds. So all of us in this room today, we're all here because we get this, we know this, and we need to tell these stories and move them on to other people so they get it and they know it and they tell the stories as well. And we need to be able to articulate the importance of the megafauna, what I'm calling the pulse of the Gulf, because if people care, they will protect it. Thank you.